Okay. Hello, everyone, and welcome back to the Swalife Podcast Series. In drug discovery, there's a moment every researcher celebrates the moment a molecule shows strong biological activity. It feels like the hard part is over. But in reality, this is where many drug programs quietly begin to fail. A significant number of promising molecules never reach patients, not because they don't work biologically, but because they cannot be formulated into a reliable medicine. So, today we are exploring a critical and often underappreciated question: why do formulations fail after discovery? To help us unpack this, I am joined by Dr. Pravin Badhe, who has worked across discovery science, formulation strategy, and translational development. Welcome back, Dr. Pravin.
Thank you, Shruti. This is a topic actually every scientist should understand before actually understanding why formulation fail is one of the most common and costly reason the drugs is actually disappearing from the discovery.
Yes, so let's begin with fundamentals. When people say a formulation failed after discovery, what does that actually mean? Is this a failure of the molecule or of the development process?
Okay, so actually it's nice question to begin with. That distinction is crucial actually when we look at the formulation failure. Formulation failure rarely means the molecule is inactive or poorly designed. Actually, it means that the molecule cannot be converted into a form that behaves predictably in humans. So, in practice, formulation fail can mean that the drug does not dissolve adequately in the body. It degrades during the storage or transport. It interacts negatively with the excipients. Now, that is another important part of the formulation. Then it cannot be manufactured consistently at the scale because most of the time large scale, low scale manufacturing is done, or it fails to deliver sufficient exposure in patient. So basically, the molecule may work perfectly in all these assays in test tube, but medicine does not live in the test tube, and that's the reason sometimes the molecule is working, but the formulations are not working.
So, is formulation failure really a translational failure?
Exactly. It's the failure to translate chemical potential into clinical reality. So formulation where chemistry meets biology, physics, manufacturing, and patient behavior all at once. So that's important part.
If formulation is so central, why are these risks often missed early on?
Because discovery and formulation are optimized for very different goals. So when we look at discovery science asks, does this molecule hit the target? Where in formulation science, the question is, can this molecule survive in the real world? So during discovery, molecules are tested in artificial solvents at concentrations that don't reflect human dosing. Then under short terms or ideal conditions are used in the discovery. But with the formulation, there are real world tracers which actually making sure that the formulation is formulated for water-based environment and the temperature humidity changes that the environments are being utilized and long-term storage is happening with the formulation and mechanical trace during the manufacturing is also happening which are not there in the discovery because these factors are not immediately visible in early screenings. Formulation risks accumulate quietly and only explode later.
That was a very well explained answer. So let's go deeper now. Which molecular properties most commonly sabotage formulation success, and why do so many potent molecules struggle at this stage?
Okay, so if we look at the molecules, the biggest culprit is the poor solubility. So many modern drug molecules are designed to bind the target, which often make them large, lipophilic, and poorly soluble. So other major contributors, including chemical instability over time, sensitivity to moisture or light, then polymorphism and crystallization behavior, then pH dependent degradation. So all these properties may not affect in vitro assays, but they completely determine whether a drug can be delivered reliably in the humans.
So can the formulation strategies always fix these issues?
Not always. Like advanced formulation strategies, like nanoparticles or lipid system, they can actually help, but they actually add the complexity. Also, there is a cost involved, and also the regulatory risk is increased when these advanced formulation strategies are implemented. At some point, the question becomes: are we actually rescuing a good molecule or forcing a bad candidate forward?
Potency often looks excellent early on. Why does bioavailability later become such a critical bottleneck?
Because the potency and the exposures are fundamentally different. When we look at these two areas, discovery focus on how strongly a molecule interacts with the target, where formulation determines how much of the molecule actually reaches to systemic circulation. So many drugs fail because they actually have they dissolve too slowly, or they are metabolized before absorption, or they show high variability between individuals. So when exposure is inconsistent, clinical outcome become unpredictable, and that's undermine confidence in both efficacy and safety. So bioavailability is not a formulation detail; it's a clinical determinant.
Okay, so the next question is people often call excipients inactive. How misleading is that term?
Uh, as I mentioned earlier as well, that the excipient is part of the formulation, and uh, calling it as inactive, it's very misleading because excipient can actually accelerate the degradation. They can alter the drug release profile. They can also affect the absorption kinetic, and they can create stability problems as well while for the when the drugs are stored. So on top of this, formulation that perform well at lab scale often fail during the manufacturing. Why? Because there is changes in mixing intensity, compression force, batch size, and also the equipment type, which can actually dramatically alter the final product. A formulation that cannot be manufactured consistently is not a viable medicine. So no matter how good the science looks.
This is often overlooked. How do patient-related factors cause formulation failure? Can a scientifically sound formulation still fail clinically?
Okay, so that's another main important part, because this happen often that people realize the formulation is failing. And formulation fail when they require too much frequent dosing, have poor taste or odor, are difficult to swallow, and also require complex administration steps. So the patient behavior plays important role. So if patient cannot or will not use the medicine as intended, real world effectiveness of that drug can actually or the formulation will collapse.
So is formulation also a behavioral science problem?
Yes, formulation success depend not just on molecule but on human behavior as well. A medicine must fit into the patient life, routines, and limitation, or it will fail silently after the launch of that formulation.
We are towards the end of this episode. So, to close, what lessons should scientists take from these repeated failures?
So the biggest shift is the integration. The formulation should not be treated as a downstream rescue operation. It must be part of the early strategic decision making. So at Swalife, we actually do that. When we have the discovery done where leads and targets are binding, simultaneously we go for formulation like capsules, tablet, and we make sure that it has been used at the early stage. So the key mindset change include evaluating developability alongside potency. So just not looking at the potency, but also need to look at how we can develop that into the formulation. Identifying formulation risk early, even if it is slowly progressing. Then encouraging discovery and formulation team to actually co-design the molecule, thinking in terms of the clinical use, not the patient use, not just the laboratory success. The most common question is not that how the molecule work or can we actually make this molecule work. It should be, does this molecule deserve to become a medicine? So that should be the important question.
Yes, so that's a very powerful way to think about this. Today's conversation makes one thing clear that formulation failures are rarely accidents, they are often delayed consequences of early decisions. Thank you, sir, for breaking this down so thoughtfully.
Thank you, Shruti. If discovery is about possibility, formulation is about responsibility. So we need to think about it in that way.
And this is to our listeners. If this episode changed how you think about drug development, share it with someone who needs to hear this. And join us for the next episode. Thank you.